Good morning, everyone. Thank you. So given that company presentations are relatively short, I'm going to move through this pretty quickly, really focusing on the evolution of our company since we last spoke last year. So Caladrius is a public company, and I will be making some forward-looking statements throughout this presentation, and I'll ask you to keep those in mind if you intend to make investment decisions based upon what you hear this morning. So as you probably recognize, historically, Caladrius operated a hybrid business model. We owned PCT, a cell therapy manufacturer, and as well as developed therapeutics on our own. And over the course of time, we came to the conclusion that being able to adequately advance and optimize both of those businesses simultaneously was going to be a real challenge. And so we've, in fact, recently completed a business model evolution which allowed, and I hope you can see the cell mitosis happening in front of your eyes here, the separation of the two businesses into two independent entities, with Hitachi Chemical now being the sole owner of PCT and Caladrius being the sole owner of our development programs. This allowed two companies to emerge that are healthy to go forward. It provided Caladrius with much needed capital for growth on the development side while preserving the very intimate relationship we have with our manufacturing partners at PCT, and in fact allowed us to negotiate as part of the contract some discounted fees for some of our cell therapy platforms, which in fact puts us in the ironic situation of paying less for manufacturing as a customer than we did as an owner. So this puts the company in a very new position. It's focused, it's poised for growth, and for the first time in quite a while, it is actually very well funded. We are a pure play, clinical stage therapeutics development company with two technology platforms on which we are currently building. One uses polyclonal T regulatory cells in immune modulation, and the other uses CD34 plus cells in ischemic repair. We have approximately $60 million of cash on our balance sheet, several years worth of operating cash. We are also debt-free, having paid off all the accumulated debt that the company had from previous management. And we are in the middle of an ongoing landmark phase 2 study of our T cell technology in diabetes, as well as about to start a phase 2 trial, which will actually be eligible for early registration in Japan in critical limb ischemia. And I'll speak to both of those programs in more detail now. So in immune modulation, we're working on autologous polyclonal T regulatory cells, and we're looking at this as an application for autoimmune diseases. The technology is based very soundly on a platform built by Jeff Bluestone and his colleagues at the University of California, San Francisco, as well as some IP from the Centenary Institute. As I mentioned, this is an autologous, ex vivo expanded and activated polyclonal T regulatory cell approach to treating autoimmune disease. We have exclusive rights internationally to a portfolio of issued and pending patents, and thanks to our colleagues at PCT, we have a well-characterized and optimized proprietary manufacturing process, which is resulting in cost of goods which are very, very manageable. So the approach here is very straightforward. Most people believe that in autoimmune disease, there fundamentally is an imbalance between T effector cells and T regulatory cells, either an insufficient number or insufficiently potent. T regulatory cells allow the T effector cells to attack beneficial tissues in the body. In the case of our target initial disease, type 1 diabetes, your T effector cells attack the beta cells and ultimately reduce the body's ability to produce insulin. Our goal here is to augment, both in number and potency, the T regulatory cells that are naturally occurring within the body, with the intention of restoring the immune system to its native state of balance and reduce and hopefully ultimately eliminate autoimmune disease symptoms. This approach, we believe, is applicable across a wide variety of autoimmune diseases. You can see a list of them here. This is not meant to be all-inclusive, but it shows that the market potential here for this approach is quite large. And, of course, the same approach from a product perspective could be generally applied across the board. We have chosen to start in type 1 diabetes for our proof of concept. It's a historical decision, and it is a market that is largely unmet. It has approximately 20,000 newly diagnosed patients under 20 per year. There are no curative treatments, only lifelong insulin therapies, and there are many, many known comorbidities associated with a long life with type 1 diabetes. Our goal here is to preserve remaining beta cell function in patients newly diagnosed, that is with recent onset of disease, with the expectation of slowing or stopping disease progression and leading to a much more normal life and to long-term medical and pharmacoeconomic benefits. So our manufacturing process is quite straightforward and is much less intrusive than maybe some other cellular therapies. It does not involve any bone marrow aspirations. This is a whole blood collection. It is about 14 days from donation to patient infusion, and the process is, as I said, well characterized and very reliable. We are approximately we're very close to the end of patient enrollment in an 111 patient study and have had very few manufacturing failures, so we believe that the reproducibility here, or the reliability rate, is well above 95%. So this trial is very interesting in that it is the first trial that is placebo-controlled, randomized, and studying patients in this population using this technology. That's why we call it a landmark phase 2 trial. It's known as the T-Rex trial. It is funded by a number of research partners. Notably, CIRM has given us a grant of up to $12 million to support this trial. JDRF has provided some money to help support the immunoassay portion of the analyses, and our strategic partner, Sanford Research, has made an equity investment in our company in support of this trial as well. The program has a number of regulatory accolades. It's got FDA Fast Track and Orphan designation, and that Fast Track designation is the first time FDA has given that categorization to a type 1 diabetes program. We also have ATMP designation or classification in the EU. And our approach fits very nicely, we believe, in the paradigm for treatment of diabetes. On the far left, you have the sort of the Big Pharma approach, which is a chronic approach to treating disease, symptom management, improved ways to deliver insulin, new insulin analogs, new delivery systems, etc. And these are sort of readily available in development and coming on the market. On the far right of the slide, you have the future, which is a purely regenerative approach, teaching either new cells within the body to produce insulin or somehow regenerating the defunct beta cells. We believe that that's sometime in the future and probably a ways away. And so what we're looking at is a cell therapy treatment that's squarely in the middle here, which is designed to reduce or eliminate progression of the disease, allow the patients to live quasi-normally with their existing functioning beta cells in anticipation of a regenerative medicine cure at some point in their lifetime. The study is based on some very sound and exciting phase 1 data done in the United States and in the EU, where it was shown that the therapy is safe, where the cells have been shown to be durable and have a potential for a reliable effect, and in an EU open-label study where it was demonstrated that there could be a fairly substantial early therapeutic effect with the ability of even reducing or eliminating the need for insulin in some pediatric patients. So this has led us to the T-Rex trial, which, as I said, is double-blinded, placebo-controlled, and randomized. This is a trial being done in the target patient population with the standard regulatory and medically relevant endpoints, powered appropriately in terms of size, and really studying several doses of cells based on a single infusion to get a proof of concept and to, as most phase 2 studies do, to define and inform us on the next steps in development. The timeline for this trial is such that we are approaching the end of enrollment. We had predicted that we would complete enrollment by the end of this calendar year, and we're well on our way to doing that. We've also reached a number of milestones, notably the 50% of subjects treated milestone back in the early part of the summer, which started a 6-month clock to a prescribed interim analysis, the results of which will likely be announced in the mid to late first quarter of next year. The 70th subject triggered a capital infusion based on a commitment from investors made in September of 2016, and we'll be moving forward collecting milestones from CIRM based on enrollment and study execution as well. So we're about a quarter away from announcing the interim results and about a year away from reaching the primary endpoint in the study, which is 1-year follow-up on all patients. So now I'll move quickly to talk about the other platform, which is using CD34 cells in ischemic repair. This is a platform that is based upon what we believe is a profound body of clinical evidence that demonstrates that CD34 cells do have a therapeutic effect in restoring microcirculation in areas that have achieved or experienced an ischemic insult. Much has been done in the way of clinical as well as preclinical studies. There are a number of references here, and we believe that opportunities exist across a multiplicity of disease states, but we're focused initially on critical limb ischemia in Japan, where we're taking advantage of the regenerative medicine accelerated path to approval, also coronary microvascular dysfunction, and ultimately refractive angina. The study that we're about to embark on in Japan in CLI is a phase 2 protocol, and we've had that protocol as well as our CMC strategy completed in consultation with the Japanese PMDA. This is a study that we've been told, if successful, will be eligible for consideration for early conditional approval. It is prospective, open label, controlled, and randomized in patients with no option CLI. It's open label because it's very difficult to enroll patients in a placebo-controlled trial where the folks on placebo are likely to see amputation as the result. And so we've had agreement to do an open label trial. We also have what we believe is an advantageous endpoint. The primary endpoint is time to continuous CLI-free status, defined as 2 consecutive CLI-free months, and these are monthly visits. So with patients beginning enrollment later this year, we could have results on the first set of patients as early as the beginning of the second quarter of next year, and since it is open label, we'll be able to talk about that. We're using intramuscular injections, 20 injections in the affected lower limb, and as I said, the first patient is targeted within the next quarter. We also have recently received a NIH small business grant to support a pilot trial in proof of concept in coronary microvascular dysfunction. This is a $2 million grant that will fund a 20-patient proof of concept trial that'll be done here in the United States in concert with the Mayo Clinic and also the folks at Cedars-Sinai, UCLA. It's a phase 2 proof of concept trial. First patient in will probably be in the early part of the second quarter of next year. And since it's a 6-month endpoint in this trial, this is also something for which we expect to have results to discuss in 2018. So you can see our pipeline has broadened considerably since last year. Our type 1 diabetes trial is nearing the completion of enrollment. We have a partner in Jeff Bluestone who's doing some investigator-initiated work in kidney transplantation and cutaneous lupus. The results of that will inform us on next steps in our own development program. Those are indications that are part of the license to which we have access. And then our CD34 program, by this time next year, not only will have had 2 clinical programs running, but likely we'll be reporting results on both of them. We have, over the course of the year, had a number of newsworthy events, value-creating milestones, and we predict to achieve a number more of those during the course of 2017 and through 2018. I won't read you the list, but as I've said, we have data coming out for an interim analysis on CLBS 03 in the type 1 diabetes trial. We have the initiation and then ultimately data reports on the CLL trial for the CD34 platform, CLBS 12 during the course of 2018, and we also have the CLBS 14 program in coronary microvascular dysfunction, which should initiate next year, and toward the end of next year likely be around the time that we'd be seeing the first data from the endpoints in that trial as well. We've closed the sale of Hitachi PCT to Hitachi, which brought into the company almost $80 million in revenue. That has funded us now for the next several years and given us the opportunity to not only affect the business plan that I've just discussed, but perhaps even consider adding to our pipeline appropriately going forward. So a lot has happened with our company. We've really evolved over the last couple of years, reached a number of milestones, have a much stronger balance sheet, and 2018 proves to be a very interesting year for us from the data perspective as we move our programs forward. Thank you very much for your attention this morning. I think we have perhaps 1 minute if anyone has a quick question, and then I'll yield the podium to the next speaker. So thank you for your attention.